Good morning, everyone. On behalf of the GCC Innovative Drug Discovery and Development Consortium, I want to welcome you to our second annual ADME Lead Characterization and Optimization Conference. I know that we are all in for a great treat today. I want to first of all give a shout out to CPRIT, who through funding of the GCC Center for Comprehensive PK, PD, and Formulation at Texas Southern University, are actually funding this conference today. Of course, they've done so much more for the cancer therapeutics landscape in the TMC and across our GCC institutions. Next slide, please. All in for a great treat today. This slide gives an overview of our CPRIT-funded core network that includes a lot of sophisticated core equipment and expertise that span the drug discovery and development pipeline. I've highlighted CCPF, or the Center for Comprehensive PK, PD, and Formulation, which you'll hear a lot more about throughout the course of the day. Next, I want to acknowledge Texas Southern University College of Pharmacy and Health Sciences. So much to say about these folks, but for the sake of time, I will mostly let their knowledge, expertise, and the research that you'll hear about throughout the day speak for them. I would, however, be remiss if I did not tell you that this group is exceptionally talented and one of the nicest groups I've ever worked with. I've had the privilege to collaborate with many amazing and brilliant scientists over the years, and I can say without hesitation that I have enjoyed none any more than I have our collaboration with this group. With that, I would like to introduce Dr. Dong Liang, the director of CCPF and the interim associate dean for research at TSU, Dr. Diana Chow, who is the Paula and John J. Lavoie Endowed Professor in Drug Discovery and Development, also Professor of Pharmaceutics and Director of the Institute for Drug Education and Research at the University of Houston College of Pharmacy, and Dr. Huan Xie, Professor and Co-Director of CCPF. And these three will now give you a little bit of an introduction to CCPF.
Good morning. My name is Huan Xie. I'm the co-director of CCPF. Thank Suzanne for the wonderful introduction of our core. This core full name is GCC Center for Comprehensive PK, PD, and Formulation. CCPF actually provides preclinical drug development service from pre-formulation to PK/PD modeling, which includes wide variety of in vitro, in pseudo, and in vivo study described in this scheme, with 9 specific areas, as you can see. The one I'm responsible is pre-formulation and formulation. Pre-formulation characterization is focused on drug stability and solubility. We have a lot of advanced instrument in our core facility, which can provide a comprehensive characterize of the drugs. So for example, we have the PION Ceres T3 system, which is capable of measuring physical chemical properties, such as pKa, pH solubility profile, oil-water partition coefficient like P, and drug solubility in various types of solvents. We also provide services on developing pharmaceutic dosage forms, such as co-solvent, microemulsion, etc., which is suitable for in vivo studies so the investigators, our user, can move forward to their animal studies. And for those non-conventional compounds, sometimes very difficult, very insoluble, we also develop more advanced drug delivery systems such as liposome, solid lipid nanoparticle, polymer conjugates, nanosuspension, nanoemission, etc. So we have full capacities to doing those formulation and characterize the drugs. Next, Dr. Liang. Dr. Liang.
Unmute.
Preclinical pharmacokinetic evaluations generally involve 3 areas. First is to develop a sensitive and a specific bioanalysis to quantitate drug concentrations in the plasma, blood, or tissues with a limit of quantification down to as low as nanogram per milliliter range. Our GCC center has 4 LC-MS systems running 24/7 for this purpose. The second is to evaluate in vitro pharmacokinetic characteristics of a drug candidate. And this involves hepatic drug metabolism studies, the CACO2 cell permeation studies, and transporter studies, protein bindings, as well as drug stability in plasma. We also have in situ gastrointestinal permeation rat models for drug absorption studies. And third area, of course, is to conduct in vivo pharmacokinetic studies using rodents as animal models. Mostly we're using rats and mice. And those studies include crossover oral bioavailability studies, dose linearity studies, IV infusion and multiple dose steady state studies, and tissue distribution studies. In this slide, you can see that these are examples of pharmacokinetic evaluations, such as rat microsome metabolic studies, the in situ GI permeation rat model studies. The PK plots in the middle of this slide is an example of a dose linearity study using rat as the animal model. And we characterized a cancer and anti-cancer drug, prodrug, oncosan, its active drug, and its active metabolite at 3 intravenous dose levels to verify dose proportionality, and which is a required PK steps in the drug development. And Dr. Chow will introduce the PK/PD modeling.
Okay, we are in the last section of this core facility. However, that's definite, also very important. We provide those consultation on experimental design for those IND enabling preclinical studies, and also would do the comprehensive PK modeling, development, and simulation. And third is the PD modeling and determination of those parameters. And lastly, the PK/PD modeling analysis that help us several aspects. One is to design the experiment for a clinical trial and decide the first dose in human, and also can rationalize the observation in clinical studies or clinical trials. This slide is showing different areas of services we have at UH. We have that expertise, personnel, and also advanced computer PK modeling on top of those 4 boxes. And the experience we have is quite intensive. We have studied those intranasal scopolamine for NASA, and also we're co-modeling parent drug and metabolites. And also we have a model to describe very complicated process like an enterohepatic recycling. And for PD, we have the experience to monitor the maternal and fetus PD. And lastly, the PK/PD correlation with the statins that's in the study for patients, clinical study after the gastro bypass surgery. And we found that for those LDL suppression or decrease, not only depend on the drug dose, also that's a progressive model of the BMI. So we're also dealing with the modeling with the disease model. Okay, without further delay, I would give the time to interim associate dean of TSU, Dr. Omeneki Olaleri.
You got it.
Yeah, thank you.
Thank you, Dr. Chow. Thank you. So welcome again, and Texas Southern University is happy to host this important conference with the GCC. We're really happy. It's been a great collaboration with Dr. Tomlinson, as she mentioned earlier. And I bring you greetings from our President Ken Hewitt, as well as our Provost Kendall Harris. We really enjoy this collaboration. We're playing a key role by providing the services not only to the Texas Medical Center, but also to the state of Texas. And at Texas Southern University, I just wanted to give you a few facts from our university. We have about over 700 faculty with close to 8,500 students. And we're one of the largest historically Black college and universities in the country. And actually, Texas Southern University, we're always proud to say that we're #1 in producing a lot of the underrepresented minority undergraduate and graduate degrees, even in the state of Texas. Next slide. We have 10 colleges at Texas Southern University: our College of Science, Engineering, and Technology, the College of Pharmacy and Health Sciences, our School of Public Affairs, our School of Communication, our College of Liberal Arts and Behavioral Sciences. We also have our Thurgood Marshall Law School, the Jesse H. Jones Business School, as well as the College of Education and the Honors College and Graduate School. Next, please. And we do pride ourselves, like we said, strategically positioned at the largest medical center in the world. And we've been able to leverage our strengths and partnerships with our collaborators. We have a successful PhD program in our pharmacy school, as well as in the College of Environmental Sciences as well. Next slide, please. And this slide is just to show you one of our major initiatives at the university in terms of using a synergistic approach to discovery, specifically more so drug discovery and development, where we've identified novel therapeutics, novel targets. We have a very strong team that, as you're going to see today, in our Center for Comprehensive PK, PD, and Formulation, which was funded in 2018 by CIPRID for preclinical drug development of anti-cancer drugs. We also take pride in educating and increasing the translational workforce and pipeline, as well as novel maternal and child health programs. We have 2 major MCH programs. One of them was recently funded from our distinguished professor, Dr. Bullard, in collaboration with Baylor College of Medicine by by the NIH, $7.1 million grant. And more recently, we received what we call a RCMI grant, a Research Center for Minority Institution grant, $8.63 million from the NIH MHD, to actually play more of a strategic role at the medical center, where we position ourselves to bridge the gaps between our community as well as the advancement of science and clinical drug development at the medical center. And we also more recently started to play a key role even during this pandemic, where we've identified potential therapeutics for antiviral therapies, and we're collaborating with Emory University School of Medicine to begin phase 2 clinical trials. So we're really excited at this time, and I hope you enjoy this conference, where you hear more from our faculty and collaborators that have used our Center for Comprehensive PK, PD, and Formulation. Thank you.
Okay, we'll give the time to Dr.
Yes, I think the next speaker, we're actually going to session 2, formulation and PK, continuing this wonderful morning. And I will be the convener for this session. And our first talk is from Dr. Chai, Dr. Liu, and myself on the topic of supralingual delivery of mycophenolic acid, PK analysis and modeling. Just a little bit background, Dr. Tsai is the associate professor at the Texas A&M University Health Science Center, Institute of Biosciences and Technology, who is the driving force of this wonderful project. Dr. Xiaohua Liu is associate professor at Texas A&M University College of Dentistry, who designed and synthesized a 3D printer on this novel transmucosal patch. And myself is a professor of pharmaceutics at TSU. We, in our core facility, where we perform the in vivo PK evaluation, tongue distribution, and sample bioanalysis with LCMS instrument. So without further ado, Dr. Chai, you want to share your screen on this presentation?
So am my screen being shared right now and everybody can hear me?
Yes, perfect.
Okay, so, well, thank you, Dr. Xie, for your introduction. And I also echo Susan's comment on my experience in collaborating with the PKPD core in terms of the productivity in science as well as the pleasure in personal interaction. So, and that's a very productive, constructive experience for me. And as Dr. Xie introduced that this presentation is going to be done collaboratively, like we have done in science by the three of us. And so I'll be driving the flow of this presentation, but at appropriate time, Dr. Liu and Dr. Xie will be presenting their part of the projects. So, so the starting point or the initiation's idea for this project is that we're trying to develop a non-invasive oral delivery device to deliver drugs to treat oral precancerous lesions, also known as leukoplakia, as a prevention for oral cancer. So oral cancer, although it's not at the top of this cancer prevalence chart in the United States, it still accounts for the 8th most common cancer in men and the 15th most common in women. But the worst part of the oral cancer is that this 5-year survival rate is pretty low. It's 63%. And worst of all, the treatment for oral cancer often results in severe complication, including systemic side effect if it's a chemotherapeutic agent, or severe deformity in the oral cavity that affects not just the appearance, but also the speaking or drinking or eating function. So the life quality is severely affected. And the best way to treat the disease that has high mortality and morbidity would be to prevent the occurrence before they become cancer. So a silver lining of oral cancer is that the majority of oral cancer belongs to this oral squamous cell carcinoma, and most, if not all of them, are believed to develop from precancerous lesions that are clinically recognizable as leukoplakia, shown in this the panel A and the panel B. So there are two types of oral dysplasia. One is low grade, the other is high grade. For the low grade dysplasia, the malignant chance is about 4 to 10%. For high grade dysplasia, the chance increased up to 30%. So for management of high grade dysplasia, it is well accepted that it need to be treated aggressively by surgical removal or by laser ablation or by chemotherapy. Because of the high risk of becoming cancer, that justified the risk of developing severe complication. But it's a different story for low-grade dysplasia, which only has 5% of becoming cancer, 5% of chance of becoming cancer. So therefore, that become a dilemma for the physicians to decide whether we want to treat the 5% chance of becoming cancer at the 100% risk of developing severe complication, especially for lesions that affect the whole surface of the tongue, such as the B panel B, showing here in this slide. So it's almost impractical to do laser or surgical treatment for lesions that are diffuse or multifocal or recurrent. And if we don't, then there's still a significant minority of those patients that will become cancer. So far, there's no good treatment for this group of patients. And that's what we're trying to do here, is to develop a non-invasive drug-based local delivery device to treat those diffuse or multifocal low-grade oral dysplasia and to eradicate the lesion to prevent them from becoming cancer. The drug side of this design is based on the synergistic interactions that, in a way, kind of similar to what Dr. Huang has described, the combinations of drugs. And here we found that the mycophenolic acid or mycophenolate can act as a non-chemotherapeutic agent chemosensitizer to sensitize multiple different chemotherapeutic agents in killing various different cancer types. And in relevant to the focus of this presentation, I'm showing here is the ability of mycophenolic acid in combination with 5-FU polytaxols or oxaliplatin. That's also one of the drugs that Dr. Huang has mentioned in killing either oral squamous cell carcinoma cells or killing the dysplastic precancerous oral keratinocytes in cultures. And what we found here is that by combination with oxaliplatin, mycophenolic is able to achieve a high synergistic activity based on the isoborogram calculated by a combination index below 0.3. And here in this cancer is 0.2, and in the dysplastic cells is even better, is the combination index is 0.02. So the next step is to how to deliver this mycophenolic acid onto the oral mucosal tissues. So for that purpose, I collaborate with Dr. Liu at Baylor College of Dentistry to formulate a novel mucoadhesive patch. So I will let Dr. Liu to introduce the part of the work that he has done to formulate the oral mucosal patch. So Dr. Liu.
Okay, so I talk about the topic patch design. So we know that for topic drug delivery, we can have different formula. We can use film, template, or hydrogel, or even beads. But for this specific project, we want our drug specifically target on the mucosa side, but not on the oral cavity. So based on that, we choose to use the patch design. So our patch design will have a layer by layer structure. As we show the cartoon in figure C, you see that we will have first of all, we'll have a back layer. So that back layer will prevent any liquid to penetrate. And then the next layer we will have the drug layer, and then also the combining with our bioadhesive materials. So we use polyacrylic and also CMC cellulose. So in that way, the drug and the bioadhesive material will be able to mix together and then also attach on the mucosa side. And for the patch fabrication, we use the 3D printing technology to do that. Where we think that we use the 3D printing, we can better control the combination, the amount of drugs. Also, we can get very easily to control the thickness of the patch. So our patch, we can use this way, we can control the thickness to just a few hundred micrometers or even thinner. So the next slide. So this one is we used the machine to test the mechanical property, especially in the bioadhesive strength. So we target on the patch. When the drug is released, it can stay on the mucosa side for a specific time point. But after the drug is released, it will be easily to be peeled off. And we targeted the bioadhesive strength about 1 newtons. So in that way, we can easily peel off and not it to induce the irritation. So we screened the different formulation, and later on we found that the optimal combination can really achieve our goal, that is to get the peel force about 1 newtons. So so that's the bioadhesive patch design, and I will stop here.
Okay. So after rounds of optimization based on in vitro data, we decide that the patch is at the stage that we can test it in vivo in preclinical models, i.e. rat, spray dolly rats. So the first thing we test is the in vivo mucoadhesiveness on the mucosal membrane in the rats. So we tested 3 locations. So here in this image, the patch is sized as either 4 millimeter or 3 millimeter circles placed on the anterior part of the rat tongue or posterior part of the rat tongue or on the buccal membrane. And we put the rat to sleep for a number of hours, from half an hour to 4 hours. And what we found is that regardless of the size of the patch can stay on the mucosal almost 100% throughout the period of 4 hours, as long as the rat is asleep. And we also test the stability of the mycophenolate in the patch format. And we kept the patch at 3 different temperatures. One is the -20, the other is 4 degrees, and the other one is room temperature for 1 day, 4 days, 7 days, up to 6 months. And we're actually currently testing it at 9 months and 12 months. The 9 months will come up actually tomorrow. So at least for the results that we have tested up to 6 months, the mycophenolic has been stable regardless of the temperature, as much as a stable by comparing to a freshly made patch. So the next experiment, the next few experiments would be to test the in vivo efficacy, the in vivo toxicity, which as well as the pharmacokinetic analysis in vivo. So for the focus of this symposium, we're going to talk about just the pharmacokinetic analysis, which is critical for having this device be approved by the FDA for clinical trials. So for the next 6 slides, we will have Dr. Xie to talk about the PK analysis. Dr. Xie.
Thanks, Dr. Chai and Dr. Liu. From the wonderful introduction, we can already see the exciting new technology for the local treatment of oral cancer with many potential advantages, such as the high dose accumulation at the targeting site, and also the low toxicity because very limited amount of drug will go into the blood. So, but there's, when we first get this project, there's only very limited reports on transmucosal delivery, especially on the PK pharmacokinetic study. So the challenge in front of us is how can we approve this novel non-invasive patch can really do what we expect it to do. So we designed 2 sets of experiments to answer the question. The PK analysis is the tongue tissue distribution. As you can see from this chart, for both of study, we'll apply patch on top of the tongue on rats for 4 hours under anesthesia. For the PK study, which is the left-hand side, we started with blood draw right before the application, and then at various time points after the dosing, up to 48 hours. So only the first 4 hours of the patch applications under anesthesia. After that, the rat is free to move. So this is actually 44 hours after the removal of the patch. So it's enough time point for us to get a half-life observed. So we precede those blood samples and do all the necessary sample handling, then send to bioanalysis using our top-of-the-line LC-MS MS instrument to check the PMPA concentration in each sample. Also on the right-hand side, we did the tongue biodistribution study. Same way, we apply the patch for 4 hours. Then we remove the patch. So after we remove the patch, we wash the tongue very thoroughly. For one set of rats, we actually sacrifice immediately and collect their tongue for analysis. For another set, we let the rat live and free without drug or without patch for another 20 hours. And then we sacrifice and collect the tongue. So both the tongue tissue were homogenized and processed and also sent to bioanalysis. And meanwhile, for all the set of rats, we collect their use patch for the tongue, and also want to see how much drug is still resigned in the patch after the application. Next slide, please. So let's first look at the drug concentration plasma PK profile of the supralingual administration. So we actually did 3 different routes of administration, but this one is our focus because this is what we're interested in. Only very trace amount of the drug actually enters the systemic circulation. If you can see from the y-axis, the number is very low, 10 nanogram per ml. And you have to click, Dr. Chai, because I have a little cartoon shows. So, and you can see the peak constant, the drug level actually was not detectable until about 30 minutes to 1 hour after the patch application. And the next peak concentration was actually at 4.5 hours, which is half an hour after the patch was removed. So that shows there is 0.5 hours, half an hour of lag time from the tongue to the blood. It was then we have seen a sustained release without with constant drug concentration in the blood. Then we saw another peak at about 24 hours. And later, we see another peak at 32 hours. So those suggest the tongue is actually working as a reservoir to slow release the drug. But for the peak, is actually related to phenomenon called interhepatic recycling, which has been reported by many other literature for the MPA drug. Next slide, please. So if you look at the other PK profile we obtained for IV and oral, those are 2 traditional administration routes we're using. For those, we compare the profile. First thing I want you to notice is their concentration is much higher than that of the patch, even though they're giving the same amount of dose, 5 milligram per kg. We also notice multiple peaks in those 2 profiles, indicating the enterohepatic recycling occurs too. Can you click next and next? Yeah, so for the EHR, it actually happened when the MPA is metabolized to MPA glucuronide, MPAG in the liver before exclusion to the bile. Then the MPAG is reconverted into parent MPA by the beta glucuronidase in the GI tract and reabsorbed into the systemic circulation. So that explains why we continue to see those peaks. Next slide, please. So we have developed a very nice model to describe the PK. Here's the model for the supralingual. We also have one for oral and IV. So this is a 3-compartment model, contains a central compartment as a tongue compartment. And they were selected because they fit very well with our data. Next slide, please. So this is a PK parameter we obtained from the model with information. We can compare the IV, oral, as supralingual. You can see those data are pretty consistent, and actually the IV and oral data is compatible with the literature report. Next slide, please. So for the last slides, I want to introduce the result from the tongue tissue distribution and the patch residue. As we can see, we find the tongue has very high concentration of drug after 4 hours of patch application. Right after the patch removed, there's still over 42 microgram per gram of drug in tongue. Even after remove the patch for 20 hours, there's still over 800 nanogram per gram in tongue, which is much higher than the MPA in blood. So this shows again the tongue is a good reservoir to keep the drug and release slowly. And another interesting result is that the patch still had about 20% of the MPA left in it after 4 hours application. So this is a good information for the design of the patch and decision on the dose. So that's pretty much, I want to introduce what we have done for this project. Dr. Tsai? We cannot hear you. You probably mute yourself. Dr. Tsai, you mute yourself. We cannot. Okay. No.
No. All right. So thank you, Dr. Liu and Dr. Xie, for their contribution to this project. So we would like to acknowledge the contributions, the people that contribute, people from my lab, and also people from Baylor College of Dentistry. And also I want to mention Dr. Chen, who's an oral pathologist who also play a critical part in the study that I have not presented here, the efficacy study and toxicity study. And people from Texas Southern University, Dr. Xie, Dr. Liang's lab, and their graduate students and staff member, as well as for the PK modeling, their collaboration with Dr. Chow at University of Houston. And most importantly, we would all like to acknowledge CPRIT's generous funding for both the project itself, Early Translational Research Award, as well as for the very important PK/PD core facility at TSU by this two grant funding funding mechanism. With that, I would be happy to answer questions if we have time, or Dr. Liu or Dr. Xie will be able to answer any question.
Yes, actually, I saw one question from Ritu Gupta. This question is for Dr. Liu. The question is, can we inhibit the drug release into oral cavity and allow permeation only through oral mucosal?
See that question again?
Can we inhibit the drug release into oral cavity and allow permeation only through oral mucosal?
To inhibit the drug release.
Mm-hmm.
So here our design is not inhibited like a drug release. Our design is to control the drug to specifically release to the oral mucosa. So I'm still not very clear what that question is, to inhibit the delivery.
I think, yeah, I think
site leakage. I think it's the question is about site leakage.
The site leakage.
Right.
Okay, that's okay. If it's talking about the site leakage, that's the specific design we try to avoid. So we try to avoid the site leakage. That's what we say. We use the patch design. So we have one layer of the back layer. That back layer is specifically to prevent the drug release from the just to one direction. And we didn't show it here, but we also tried to design the honeycomb structure. The honeycomb structure at the very beginning, what we want to use is to use that structure to prevent any potential site leakage.
All right, thank you, Dr. Liu. We have one, I'm gonna give time for one more question, maybe half of that question to Dr. Tsai. While oral cancer tumor could locate anywhere in the oral cavity, why do you select the tongue for the application location? And do you think the PK would be different between different parts of tongue or oral cavity? Okay, I'll let you answer part of that.
Yeah, so, well, good question. So the lesion itself can occur at the base of the tongue, the top of the tongue, the mucosal. So it can occur everywhere. And then in terms of direct treatment, I don't see a difference in terms of direct treatment, but in terms of the PK profiles for systemic absorption, there may be a difference. And also for the tissue absorption, because on the top of the surface, it's a stratified it's a stratified with some it's keratinized stratified epithelial. So therefore it's thicker. So the absorption would be more difficult. So it's more challenging, I guess. So that's the reason we choose the top of the tongue for this paradigm. But if the lesion occurs in other part of the mucosa, then I would assume that the penetration or permeation of the drug into the tissue will be much easier. But at the same time, the pharmacokinetic for systemic absorption may be slightly different. So that maybe Dr. Xie can address that.
Yeah. So because of the limited time, I would like to answer the rest of the related question on PK and enterohepatic recycling later with maybe we can chat about that. I'm going to introduce the next speaker to keep us all on schedule. The next speaker is Dr. Ruhi Dil. And Dr. Ruhi is my collaborator as well. She is associate professor at Center for Precision Environmental Health, Baylor College of Medicine. And Ruhi and I have been working on a novel compound on its formulation and PK for cancer. And here we go. She's gonna introduce some of her wonderful talk. Ruhi, go ahead.
Thank you, Shay. You know, I would like to thank the organizers for giving us a chance to talk about this project here today. I have to say that this has truly been a team science effort. We had a lot of people from different, expertises coming together to answer the question on whether SCIP2 could be inhibited to restore nuclear P27 in cancers where P27 is otherwise oncogenic. So we know P27 is a member of the CYP family of cyclin-dependent kinase inhibitors. It negatively regulates the cell cycle. So during G1, you have high levels of P27, and this prevents the binding of CDK2 to cyclin E. As the cells get ready to progress through the cell cycle, P27 is phosphorylated and degraded, and this allows the cyclin CDK2 complex to form and for the cell cycle to proceed. Now, mutations, the abnormal expression, or even the mislocalization of P27 have been found in many cancers. And usually, a loss of P27 is prognostic of a worse overall outcome. Having just told you that P27 is a tumor suppressor that inhibits the cell cycle, I'm now about to tell you that P27 can also act as an oncogene. I've given 4 examples here of breast, endometrial carcinoma, renal cell carcinoma, and prostate cancer, where you see a large percentage of cells that have lost P27. But in an equally large fraction, we actually see cytoplasmic mislocalization of P27. And it's this cytoplasmic P27 that is oncogenic. So how exactly does P27 exert its oncogenic function in the cytoplasm? It usually enhances other oncogenic processes such as PI3 kinase AKT signaling. It increases the resistance of these tumor cells to apoptosis. It alters the microtubule as well as the actin dynamics, both during mitosis as well as during tumor cell invasion and metastases. And this is how P27 can be truly oncogenic. As you can imagine, P27 is very tightly regulated. Most of this regulation on P27 occurs via phosphorylation of various residues. And to begin with, P27 here in the nucleus, if it has to relieve its inhibition on CDK2 and cyclin E, is first phosphorylated by SRC or ABL kinase, which then primes P27 for phosphorylation by CDK2. Now, this phosphorylation on P27 at threonine 187 is recognized by SCIP2 And I have it outlined right here. So you have SCIP2, which is the E3 ubiquitin ligase, which complexes with CKS1. And as part of the SCIP called RBX1 ring complex, it actually polyubiquitinates P27, and it then gets degraded by the proteasome. However, in the cytoplasm, P27 has an alternate E3 ubiquitin ligase. It's the KPC1, KPC2 E3 ligase complex that is involved in turning over P27 in the cytoplasm. What I've also shown here is that P27 can be phosphorylated by AKT, and once it's phosphorylated at these residues, it's unable to traffic back into the nucleus. So in terms of being able to target this mislocalized P27, I think there could be two strategies to sort of do this. One would be to try and relocalize that P27 in the nucleus, and the other would be to somehow inhibit the degradation of whatever P27 we have in the nucleus so as to increase P27 levels. And that's sort of the latter is a strategy that we decided to use. The way you would sort of prevent degradation of P27, is many. You could inhibit SCIP2 itself. You could inhibit the interaction between SCIP2 and SCIP1. There's inhibitors for netylation on CULIN1, which could also be used. But all of these three approaches basically would target the entire complex, making it inactive. And therefore, this would affect all downstream targets of this degradation system. Now, this could be potentially harmful because there could be other targets which could also be oncogenic. And so the approach we wanted to take was to see if we could target the binding of SCIP2 and CKS1 to P27. So this was sort of the strategy that we wanted to use to identify lead molecules. We wanted to start with the in silico virtual screen. And this was because, just sort of starting as a fishing expedition, I think would have been too expensive and too time consuming. And so in doing this in silico virtual screen, we were hoping that we would, have lead or chemi optimization here, which would then narrow down the number of molecules that we actually tested for biological activity in a primary screen. Of course, this would then be followed by validation, as well as, hoping that we would get a couple molecules that we could put through some ADME studies, followed by preclinical animal studies, and then finally being able to apply for IND. For this, we partnered with Dr. Shuxing Zhang and his lab. Now, Dr. Zhang had shown previously using a virtual screening and in silico modeling approach. He had identified inhibitor which bound SCIP2 and prevented its interaction with SCIP1. And in this project, basically using P27, he was able to show that there's two pockets where P27 interacts with the SCIP2-CKS1. The first pocket, which I have outlined here, is basically the phosphorylation on P27 at threonine 187. This is sort of known, and this would be one druggable pocket. The other pocket is sort of new and novel, and that's where you have this glutamate 185, which binds the interface of CKS2 and, SCIP2 and CKS1. This was exciting because it gave us two potential pockets that could be, screened for targets that bound to these pockets. And basically, that's exactly what he did. Okay, this was a strategy for our virtual screening. He started by targeting these three specific interaction points. When I say three, it was basically pocket 1, pocket 2, or both pocket 1 and 2. He also utilized drug-like and lead-like chemical library, which was custom filtered for paints, to give us 5,000 hits. These 5,000 hits were further selected down to 500 using some dot conformation and other selection criteria. Ultimately, we were left with 27 structurally unique compounds which progressed to biological screening. Now, at the same time, we also screened 52,000 commercially available chemical compounds virtually. And from these, we had 24 compounds, which we passed through our primary screening assay. And the reason for doing this, of course, was to optimize our screening assay. And we were lucky because we actually found 2 compounds which showed biological activity in the screens. We call these compound 405 and compound 442. These were then sort of passed over to Dr. Gilbertson at U of H for lead optimization. So this is where compound 442 and compound 405 bind these 2 pockets that I showed you. You have compound 442 here in cyan, which is binding pocket 1, which is the threonine phosphorylation site on P27. And then you have the compound 405 shown here in purple, which binds the second pocket where you have the glutamate 185. Now, again, everything that I'm going to show you today is probably 4 or 5 years of work, which are sort of condensed into a 15-minute talk. And so, to sort of cut to the chase, both compounds were chemi-optimized, but really compound 405 was the one that we finally chose. And in doing some QSAR analysis, 3 additional compounds were chosen for the activity. I would like to point your attention to this compound right here, compound number 2, which we just called 276 for short, okay, which was predicted to be active in our screen. And I'll come back to that in just a minute. Now, for our actual biological screen, this was done as part of the Tex Act Screening Center over at the IBT under the leadership of Dr. Stefan. And here we decided to use endometrial carcinoma cells. So these are HEC1B cells right here, which we were plated on 384 well plates. 24 hours after plating, we added compounds. We did a 4-point dose curve. The cells were then fixed and stained using P27. And finally, images were acquired using an Image Express microconfocal. All of the image analysis was performed by a graduate student at the time who now has his PhD, Dr. Powell. And basically, he customized the Pipeline Pilot software to quantitate the nuclear fluorescent intensity of P27. And so what I have here is P27 in cells treated with DMSO, which was our vehicle. And then we had P27 in cells which were treated with compound 405. And as you can see, there's more nuclear localization of P27 on treatment. At the same time, they also had an orthogonal screen where cells were fixed and screened for the number of and were assessed for a number of cells to establish cell growth conditions. And all of this data was then plotted where we took area under the curve for both nuclear P27 levels as well as the growth analysis. And we had several top candidates. Now I'm going to show you again, bring your attention to this compound here, which is nothing but compound 276. This is the same compound that came out through our QSAR analysis and did indeed show biological activity, as it also fell out from our primary screen. Now we went back and we validated these by doing a dose curve over time. And these were sort of our 5 top hits that we've been working with. All of them showed increased nuclear P27. And again, to sort of cut to the chase, I am just going to be showing you data, secondary validation assays with compound 276, because this is the compound that ultimately went on for further studies. All of these secondary validation screens, though, have been done for all 5 compounds. So this was a binding assay where, of course, if this compound was indeed binding SCIP2, we should, see decreased binding between SCIP2 and P27. And this was done basically in HEC1B cells where we IP'd P27. And if you normalize for the quantity of P27 being pulled down, you actually see a reduced binding with SCIP2. Furthermore, we've also done a fractionation assay where we wanted to see if biochemically we were able to see more P27 in the nucleus. And indeed, when we treat with 276, you do see more nuclear P27. Unfortunately, over here in this particular cell line, we also saw more cytoplasmic P27, which would not be ideal. And so to see whether this was cell specific, we also did it in a renal cell carcinoma cell line called A498, where we not only see increase in nuclear P27, but thankfully we did not see an increase in cytoplasmic levels. The next assay that we've done is we've done a kinase assay. And here I'm showing you data with 405, although we're in the process of repeating this with compound 276. And you would expect that P27, if active, would inhibit CDK2, and therefore you would expect decreased kinase activity of this kinase. And that's shown right here. When you treat cells with 405, you see decreased phosphorylation on histone H1, which is a target for CDK2. We have also done viability assays where we've looked to see whether these cells are viable. Again, this was a dose curve, and here I have all 5 compounds that were chosen. 276 is shown here in the deep blue, and as you can see over dose, we see a decrease in cell viability. And we've also done an apoptosis assay to see whether we would see an increase in apoptosis with this compound. Again, 276 here is shown in orange, and we do indeed see an increase in apoptosis. Now, again, to cut to the chase, we've done this several different ways. We still have certain ubiquitination assays and other assays which are being validated as we speak. But finally, we decided that compound 276 was good enough for us to move into PK/PD and perhaps formulation studies, for which we reached out to Dr. Liang and Dr. Shea over at TSU. And I agree, these have been wonderful collaborators, very easy to work with. And I recommend anyone who's a novice at these things like myself to reach out to them, because they really do make it easy for you. We were happy to find that the compound 276 conformed to all of Lipinski's rule of 5. They went on to do, they established an analytical method for this compound. They've established solubility and stability. They've also established a formulation with a pretty high concentration of 5 mg per mil with a stability of about 2 weeks at room temperature. 2 rats were treated for sort of our preliminary PK studies. And as you can see from the graphs down here, the drug plasma levels were pretty spot on, very close to the predicted values. Now, unfortunately, because of COVID-19, we've had to kind of cut down on our animal colonies. And so we sort of paused at this state, in hopes that soon we should be able to revive animals and keep going forward with a more detailed PK study, followed by a PD study. So to summarize, we've pretty much, gone through at least, I would say, 4 steps in these, in this schematic. We're now at our preclinical ADME studies, which we hope we can take to a preclinical study. And if all works well, we hope we can apply for our IND. Now, to summarize, to acknowledge at the end, I have to be honest with you, I am just sort of an insignificant part of this project. This project was conceived over by Dr. Cheryl Walker when she was the director of the IBT, and this was done in collaboration with Dr. Zhang, Gilbertson, and Davies. This was funded by a CPRIT Early Translational Award to these 4 people right here. And these are all the other people that have sort of, stepped in and done some of the other work that I mentioned. Again, we also have CPRIT to thank for the core over at TSU, which has been hugely beneficial in trying to get this project moving forward. With that, I'll stop and take any questions.
Thank you, Ruhi. There's 1 minute left. Let's answer one of the questions. Assembling a novel library is a lot of work for 5,000 compounds. Did you start with a pre-made library? or do you have that information?
So this was actually a virtual library. So this was not a real library that was screened through the primary screen. So that's why we did everything virtually, because these are very large number of library and lead compounds, I would say, that sort of had to be curated down to a good amount. That
Computational.
Yes, so it was purely computational, correct.
Okay. One more is, was the effective concentration for apoptosis at micromolar level low enough for efficacy in vivo? That's from Dr. Chow.
You know, that's a great question, and I have to be honest with you. You know, we have it down to at a 1.1 micromole right now, which seems to be working, but that's still, I think, a bit too high. So hopefully, you know, going forward, we can sort of work out the details, mechanistic details, a little bit more to be able to sort of see if we can drop it down some.
Okay, thank you, Ruhi. Thank you for your wonderful presentation. We're going to introduce the next speaker, which is Dr. Veronica Ajawali. Dr. Ajawali is a colleague of mine who's a PharmD student, used to be a PharmD student, graduated from TSU, now assistant professor of pharmacy practice in our college, and also a board certified oncology pharmacist with clinic pharmacy practice at Houston Methodist Hospital Cancer Center, who is a rising star at TSU. So she's gonna introduce her work, combine both clinic side and our basic science sides. Dr. Ajawali, please continue.
Thank you so much for the introduction, Dr. J. Good morning, everyone, and thank you for inviting me to join in this wonderful conference today. Many thanks to everyone that is in attendance of this conference and to all the speakers that have gone ahead of me. Like Dr. J said, I'm a TSU alum, and I have the privilege to have this presentation today. So over the next few minutes, we will talk about the trends of oral formulation in cancer care, and we will focus on capecitabine, which actually happens to be, you know, the same drug, 5-fluorouracil, that Dr. Wong included in his previous presentation. So the first thing we will talk about is the trend of FDA approval of oral chemotherapy drugs. So if we look at this chart here, is a snapshot of FDA approval of chemotherapy over the past 10 years, from 2009 to 2019. And we can see that there is an uprise in the amount of approval of oral chemotherapy drugs. So 146 drugs were oral agents over the past 10 years. If we take a closer look at the past 5 years, we will see that 71% of all these drugs are oral agents. So with this information, what exactly would this mean for patients? What would this mean for our patients? And what would this mean for, you know, providers, nurses, pharmacists, and everyone that is involved in direct patient care for patients in the clinic? And then, so when we look at the practice model for the parenteral therapy, which is, you know, the intravenous route of medication administration, we would see that everything that happens regarding the care of these patients occur within a building. So within the clinic, the physician interacts with the nurse and with the pharmacist. Diagnosis, evaluation, and everything that pertains to the care of the patient is done within a physical facility. Medication preparation, medication administration, everything happens within the same facility. And also the patient comes to clinic to receive their medication. Because of that, we can easily keep track of saying, okay, we know that a patient has, you know, they are adherent to their medication. If they are experiencing any form of side effects, it's easy for us to identify what is happening to the patient. However, when we flip this same, you know, side of chemotherapy drug administration, when we flip that coin and look at oral therapy, the only thing that happens within a facility, a clinic facility, is pretty much, you know, medication, disease diagnosis and prescription that is written. Most of the time, oral chemotherapy drug needs to be sent out to a different facility, called a specialty pharmacy. And with this specialty pharmacy, there is sometimes limitation in access to medication information that is needed to take full care of the patient. Another drawback to this also is now that the patient moves out of the clinic setting, they end up taking care of themselves and administering the medication by themselves in the home of the patient. So with this, there are definitely advantages, but at the same time, we can see that we don't have that natural interconnection between medication being given in the clinic and having the patient take their medication at home. Also here, we don't have the role of the nurses, and we know that our nurses are awesome. They always help us to, ensure that multidisciplinary care of a patient. So with oral chemotherapy, pretty much those roles are eliminated and the patient takes full ownership of taking care of themselves at home. With this information in mind, we examined and looked at, okay, do we actually have, pros and cons? What are the risks versus benefits of taking oral chemotherapy? A study was done that looked at several hundreds of patients taking oral chemo and, did a survey for them, and they did highlighted that they have benefits with oral chemotherapy, including flexibility of medication administration. There is decrease in healthcare resources that is needed, as well as perceived improved quality of life, because they basically don't have to go to the clinic every week, every other week, or once in 3 weeks, depending on the cycle of their intravenous chemotherapy administration. But with oral chemo, they're at home, so there is that perceived improved quality of life. However, the risk involved with oral chemotherapy includes concerns surrounding medication adherence. Because when a patient is at home taking their own medication by themselves, we cannot guarantee that the patient will take it every day as prescribed. Maybe because of side effects, because they forget, just different things come to play when we look at having patients become responsible for taking their oral chemotherapy. Of course, another major issue to consider with oral chemotherapy is also complicated PK/PD. Sometimes take with food, sometimes take on an empty stomach, the multiple daily dosing, the complication of the regimen, 2 weeks on, 1 week off, take on days of radiation. The list just goes on, and it can definitely make the whole treatment option become a bit more complicated when the patient is in charge of taking care of themselves. Other issues also include the cost of medication, drug interactions, toxicity, as well as time to acquire the drug, from the specialty pharmacy, in between insurance and high copay that the patient needs to navigate through. So with that background information, I will just go straight into talking about capecitabine, which is the medication that we'll be focusing on today. So capecitabine is the generic name, brand name is Xeloda. This was FDA approved in 1998, and this had FDA approved indication for use in metastatic breast cancer as well as colon cancer. Mechanism of action, as clearly said by Dr. Wong earlier, is that this is a thymidate synthase inhibitor, which eventually would inhibit de novo DNA synthesis, eventually leading to cell death. So that's the mechanism of action for Xeloda. And this medication is available both in IV form. The intravenous formulation is called 5-fluorouracil. The oral formulation is capecitabine under the brand name of Xeloda. The typical dose for Xeloda will be 1250 milligrams per meter squared, depending on the indication. So it's gained a lot of off-label indication over the past several years. But just looking at a typical dosing option here, for 1250 milligrams per meter squared to be taken by mouth twice a day, typically every 2 weeks with 1 week off. And this medication is water soluble, so that means we would have low bioavailability. Because of the short half-life, it requires a multi-daily dosing regimen. So overall, we see a patient who has a body surface area of about 2 milligrams per meter squared. Such a patient would need to take 5 tablets twice a day. So that's 10 pretty huge capsules that the patient needs to take in 1 day. This definitely adds to high pill burden for the patient and can potentially cause medication non-adherence for patients over time. So with this information in mind, this basically led us to develop a study with two major study aims. One of them was to evaluate the impact of medication therapy management on medication adherence of patients on capecitabine. And the second one was to develop a PLGA formulation of capecitabine and also determine the in vivo and in vitro drug release profile for this new formulation. So the first part here is looking at the medication therapy management, which was what I implemented at the Houston Methodist Cancer Center, along with some of my collaborators at the Cancer Center at Houston Methodist Hospital. So with this, I was able to provide services, including patient education, symptom assessment, as well as comprehensive medication review, with the intention of increasing medication adherence, increasing toxicity assessment, and then decreasing the risk for drug interactions. This study was done over a 1-year period, and we then compared a group of patients that were on capecitabine prior to implementation of the MTM model in the clinic, compared them to about 40 patients that were on capecitabine after the MTM model was implemented in the clinic. And this is just showing us the basic demographic of the patients that we included in the study. When we look at the outcome of pharmacist education, pre-MTM implementation, about 42% of the patients were educated, but after this MTM model was implemented, there was an increase in patient education of up to 80%. And this, pharmacist interaction with the patient definitely led to several clinically relevant outcomes, including, increase in medication interventions, decrease in toxicity risk for the patients, increase in toxicity assessment. So reaching out to the patient to see if they have any toxicities from capecitabine, promptly reporting and promptly addressing any of these toxicities. So these are some of the clinically relevant outcomes that were impacted as a result of the study. Now, when we flip over to the basic science formulation portion of this study, so in working with Dr. J's lab in the nanoparticle lab at TSU, we were able to prepare nanoparticles for the capecitabine reformulation. Several steps were involved. The first thing, looking at process optimization as well as formulation optimization. So within the process optimization, we did a double emulsion solvent diffusion technique to be able to generate a water double emulsion for capecitabine, because we know that capecitabine is water soluble. So we had to have this technique to be able to encapsulate it in this emulsion formulation. In the process of developing or optimizing this process, two main methods were done. One was bath sonication, and with the bath sonication, we realized that there was no drug entrapment that was reported. So like we can see in NP2, we didn't have any the entrapment efficiency was nil. So then we switched that over to probe sonication at 90 seconds. We got EE percentage of 11. Then at probe sonication of 30 seconds, we were able to get 34% entrapment efficiency. Our goal is to continue to modify this process so that we can have an increased entrapment efficiency as we continue to work on this reformulation process. Then the following steps, looking at the formulation optimization, we did a couple of things looking at the effect of the pH on entrapment efficiency. So we want to boost the entrapment efficiency as much as possible while we are, changing the pH of the buffer in the solution. So at the pH of 8, we found the higher entrapment efficiency. So that's what is shown on figure 1. Then in figure 2, we try to use different polymer types to see how we can best optimize the entrapment efficiency. So the best that we've seen so far is a mix of 50 to 50% of PLGA mixed with Edrigit 1000 or Edrigit 100 at a ratio of 1:1. So with that, we found a higher entrapment efficiency ratio. Then when we look at figure 3, where we look at the effect of drug to polymer ratio on the EE percentage, we found that drug polymer ratio of 1:6 seemed to be the best at this moment. But of course, we'll continue to walk through and identify the best ratio in our future studies. Then in figure 4, we try to look at the drug polymer ratio on the effect on the entrapment efficiency, looking at Resoma polymer. So here as well, at the ratio of 1:6, we found that this gave us the best entrapment efficiency so far. So due to COVID, we had to hold off on continuing the reformulation process in the lab. So our goal over the next few weeks and months, as we safely reopen the campus, get back into the lab, will be for us to continue to optimize the process and the formulation, so that way we will be able to have a good formulation for capecitabine that would have a smaller capsule size that would will be able to, hopefully decrease the pill burden for patients and ultimately increase the patient outcomes, when it comes to them receiving this medication. Then in acknowledgement, I want to say thank you to the grants funding that I received, RCMI pilot grants 2018 to 2019 in support of this project, as well as the CPRIT core facility that provided support for some of the work that we did here. In the CCPF facility at TSU, many thanks to Dr. J, Dr. Chen, and Rita Gupta, who worked on this project. And on the Houston Methodist Cancer Center, Dr. Kirk Aini, who is one of my mentors there, as well as Tasha Roshan and Alma Mendoza. Both of them are TSU students who worked with me in the clinic to support this project. And I would open it up for questions.
All right, I received 2 questions. The first question is from Dr. Chow, who's asking, what is the stability of the double emulsion vehicles in size and drug release? I guess I will jump in and answer that question. We haven't get the chance to measure that yet because we're still in the search of the optimal formulation. As you can see, the drug encapsulation efficiency is still low at this time. So we just got some other polymers we will try because we want to improve the encapsulation efficiency at least above 50 or 60%. Now we're at the highest of 30%, which is not at our optimized range yet, but we're working towards that. And the second question is from Dr. Huang, who's asking, was the nanoparticle be given orally? If so, what would be the advantage over just free drug? So Veronica, you want to answer that?
Yes, so the intention is for us to give the medication orally. And the goal, because looking back at the slide, right now, most patients with the BSA of 2 meters squared, they end up taking 5 capsules in the morning, 5 in the evening, and that's a huge number of medications. So we hope to be able to still reformulate and offer it as an oral medication. However, maybe the dose will be reduced, so the pill size would decrease. Instead of 5, maybe patients will have to take 2 over 5, and that way we can hopefully increase the rate of medication adherence for patients.
Thank you, Veronica. Thank you for the wonderful presentation. Now let's move to the next one. The next presentation will be given by Dr. Yun Zhang, who's another colleague of mine at Texas University. She's an assistant professor in the Department of Pharmaceutical Science, and she's a mouse genetic expert who have a lot of experience on mouse cancer models by genetic engineering or xenograph. She will be introduce the PD capacity at our CCPF center. So without further ado, Dr. Zhang, are you available?
Yes, I'm here.
Okay,
Can everyone hear me?
Yes, we're here.
Okay. Can you see my slides?
Yes.
Okay, great. Oh, I guess you are seeing my slides with notes. Anyway, I'll just go ahead. Good noon, everyone. This session is about formulation and PK. I guess I will switch the gear a little bit by talking about our PD facility. It's my great pleasure to give you an overview of our PD facility at the CCPF. I will introduce you what kind of PD services that we will provide, and I will also give you an example of the cost estimate that we provided to one of our potential users, so that you will get an idea. I think it's probably somehow I can only show the presentation mode. All right.
If you want to stop the full view, just back to the original view with enlarge that. Is that going to solve your problem? You know what I mean? But it's okay this way. I think everybody can understand better with you showing the notes.
Is it? Can you see now? Oh, the same. Okay. I guess I will just continue. As you all know, the pharmacodynamics, which is study of what a drug does to the body, is an indispensable step during drug development. As an important branch of the CCPF, our PD facility uses various in vitro and in vivo assays to measure the efficacy of candidate drugs. Our in vitro assays are performed on cultured cell lines, which are either purchased from commercial vendors or provided by the investigators. And the in vitro assays that the PD facility provides include, but are not limited to cell proliferation assay, in which we use colorimetric MTT, MTS, or WST1 assays to measure effect of candidate drug on cancer cell proliferation and cytotoxicity. And we also do apoptosis assay, in which we use colorimetric caspase-3 assays to monitor for apoptosis due to candidate drug treatment. And we do DNA damage assay, in which we perform this immunofluorescent assay to measure the amount of gamma H2AX in cancer cells to monitor DNA damage due to candidate drugs. So migration and invasion are two important features for malignant cells. And if the researchers are interested in knowing whether the drug will affect those two features on the cancer cells, we can also help by performing the Boyden chamber assays to measure the effects of candidate drugs on those two features. And as I mentioned earlier, we can also provide other assays upon the request of the investigators. For example, we can measure the gene of interest by looking at the RNA expression or protein expression using the quantitative real-time PCR or Western blot analysis or immunohistochemistry. So on this slide, I want to use an ongoing in vitro study of a lead compound named TSU-01 as an example to show you how we help this investigator to explore the effect of this novel compound on the triple negative breast cancer cell line. The cell line was exposed to this compound with different concentration, as you can see, with different concentrations and for different lengths of time. As being shown here, although 1 micromole TSU-01 was not able to affect cell growth after 1 or 2 day treatments, 5-day exposure of 0.5 micromole TSU-01 was sufficient to inhibit cell proliferation and cause extensive cell death. And to further investigate the mechanism underneath this cytotoxicity effect, we also performed caspase-3 assays and gamma H2AX assays and we didn't see a significant difference between the uncultured cell lines and the cell lines treated with the drug. So this indicate that the apoptosis and DNA damage are not the mechanisms of the cytotoxicity effect of TSU-01. And lastly, we look at whether this compound would affect overall protein expression or protein stability by performing the Comassi blue staining of the protein gel, and we did not see a difference between the control and the treated group as well. So after in vitro assays, some investigators may want to go further and perform in vivo assays just to look at how the drug work inside an animal body. So our PD facility provides the in vivo assays, and we use mostly mice. And we have this xenograft model. This is an in vivo assay, and that can be used to measure growth and metastasis of cancer cells implanted in a nude mice in the presence and absence of candidate drug treatment. And in addition to the xenograft, we will also have the genetic mouse models. And we know that compared to xenograft, genetic mouse models are more superior because they have an intact immune system and therefore more faithfully mimic the clinical situation. Using the genetic mouse model, we can test the drug efficacy that develop by using the models that develop breast cancers. And we can also perform in the next few slides, I'm going to briefly introduce you what kind of genetic mouse models that we have right now in our PD facility. As many of you probably know, almost 1/3 of cancer types have a p53 mutation rate greater than 50%, and more than 1/2 have a rate greater than 30%. So our PD facility has 2 conditional mutant p53 alleles that were created and characterized by me and my colleagues when I was a postdoc fellow in Dr. Gigi Lozano's lab at the MD Anderson Cancer Center. So those 2 conditional alleles are called p53WMR172H and p53WMR245W alleles. Before any treatment, those 2 alleles will express wild type p53. However, upon the pre-recombinance treatment, those alleles will be converted and into a mutant allele that will express 2 most common mutant p53, p53R172H and p53R245W. So the wild type 2 mutant conversion of p53 actually faithfully mimic the clinical situation where how the somatic p53 mutation occurs. And it also allows us to induce p53 mutation spatially and temporally. So those 2 alleles have been fully characterized by Western blot analysis, by the quantitative RT-PCR assay, by the apoptosis assay and the cell cycle assay, and also the long-term mouse monitoring. So our PD facility can build breast cancer mouse models by using those 2 alleles. And the mutant p53 will be induced specifically in the mammary gland by mammary intraductal injection of adenovirus that expresses CRE, or by introducing the WAP-CRE or K14-CRE mouse alleles that will express CRE in the mouse mammary gland epithelial cells. So the fluorescent imaging shown here indicates the induction of mutant p53 in the mouse mammary ducts via the mammary intraductal injection of adenovirus that express CRE. And this kind of injection led to induction of the mutant p53, which led to the formation of a mammary tumor as shown here. And this tumor actually metastasized to the lung, and you can see the big nodules here. Okay. So at the bottom are HE staining of the tumors, and the pathology analysis showed that those mammary tumors developed from the mutant p53 actually have various pathology ranging from adenocarcinoma, anaplastic carcinoma, or sarcomatoid carcinomas. And this tiny figure at the corner shows metastasis in the lung, and you can see the normal lung tissue in comparison to the tumor inside the lung. Okay. And in addition to this breast cancer driven by mutant p53, our PD facility also have this transgene mouse, is MMTV new, that models human HER2 breast cancer. As you probably know, HER2 breast cancer is one major type of breast cancer, and also has a very poor prognosis. And at the bottom on the left is an immunohistochemistry image of p53 staining, showing the p53 protein got stabilized in this breast cancer. And on the right is immunofluorescence staining, also showing the p53 stabilization in the breast cancer. And we also have the intestinal cancer model, and we use this APC mutant mice. From both the unstained whole mount samples and the HE staining, you can see quite a lot of polyps being developed from the intestine, which is early stage of cancer. And at this right bottom corner, it's showing you an immunohistochemistry staining of a polyps in the intestine. So we're actually staining a nuclear protein here. Lastly, we have this well-known pancreatic cancer model by using the PDX1 CRE lock-stop logs KRAS G12D model. And you can see that large-scale pancreatic lesions developed from this model. And in the center, I'm showing you an image of HE staining showing the axillary cells and also the islets of Langerhans cells. And the Langerhans cells are stained by immunofluorescence as well. And at the bottom, we're showing the tissue-specific expression of a protein here only expressed in the pancreas, but not in other tissues. On the right, you can see that we also have this genetic mouse models for testis tumors. And you can see a comparison between the normal testis and the testis tumors. And below are HE staining showing you the anatomy of the normal testes in comparison to the testis tumors. So I guess most of the investigators will also be interested in knowing the cost. So the cost of the investigators mostly come from purchasing the cell line, and of course, depending on what kind of assays you request, the cell line, the cell culture medium, reagents, disposables, and if you prefer in vivo studies, and the cost will also come from purchasing the mouse strain and also the mouse maintenance. So here I'm using an example to show you the cost estimate. So this is the cost estimate that we gave to one of our potential users. And this investigator would like to inject an ATCC cell line into nude mice. And the time window for this study is projected to 90 days, 3 months. The costs associated with cell lines included purchase of cell lines, the mediums, disposables, and injection materials, and mouse antibody production testing, cell line authentication. And for the mouse strain, we actually gave him options of 2 kind of new mice from Jackson Laboratory, and for total 42 mice, and this is the different prices that we listed. And for maintaining all those 42 mice in 15 cages for 90 days, housing, food, bedding, and water will cost about $600. So altogether, the total cost, the approximate total cost for this investigator is $5,000. So as you can see that we don't charge for labor. We only charge for any materials that are associated with the study. Before I end my talk, I just want to emphasize that for those researchers that are interested in testing the efficacy of their candidate anti-cancer drugs, but have limited expertise or resources to perform PD studies on their own, our PD services will be invaluable. So the PD facility is overseen by me and Dr. Engelby Martin. And if you have any questions or if you're interested in using our facility, you're welcome to contact us. And you're also welcome to visit our website listed here to view the comprehensive list of services offered by our facility. Last but not the least, I would like to take this opportunity to show our appreciation. We thank CPRIT for funding our facility, for funding the core. And we also thank the RCMI grant. We used the molecular and cellular biology core to perform the study with the lead compound. And last, we also thank Dr. Audrey Player for providing the cell lines for the TSU-01 efficacy studies. With all this, now I will be happy to take any questions.
All right, thanks, Dr. Zhang. So I think the audience probably can type the questions through the chat. If you are interested in our PD service, feel free to contact any of our director or contact Dr. Zhang to know more about the specific service we provide as the CPF. And now we're gonna have a lunch break / virtual poster session. We have about 22 wonderful poster presented by researchers from TMC. And you are feel free to join any of the poster on the umbrella. You can actually see them through the schedule. You just need to click into the link and find the one you're interested in. And you can visit multiple of them, of course. So we're gonna start at 12:15, which is in 3 minutes for the posters, which last until 12:55. And our afternoon session will start at 1:30. Correct me if I'm wrong. I believe the afternoon keynotes start at 1:30. So with that thing, thank you for the morning session, and we'll come to our poster session now.
Okay, we're going to get started for the afternoon section. Indeed, our great pleasure that we can have Dr. Stephen Schmidt to be our keynote speaker for the afternoon section. Dr. Schmidt has very intensive training from Germany, and then a PhD from Florida, and the postdoc from Amsterdam. And I first recognized him as a rising star in USA in the field of pharmacometrics and also systems pharmacology. But just a few years, he has very well established himself as a faculty and also strong researcher. And several of our PhD graduates received the postdoc training from his group, and very well landed in major pharmaceutical company with this expertise. So today we really expect to learn a lot of insightful experience on challenges and opportunities of a model-informed drug development. Without further delay, I give the time to Dr. Schmidt.
Thank you, Dr. Chow, for the very kind introduction, for the very kind words. And it's my pleasure to be here today. And thank you for also giving me the opportunity and inviting me to speak to you. So before we go into the actual presentation, I would like to outline 4 goals for today's presentation. First, I would like to provide an overview of the drug development and regulatory evaluation process. Second, I'd like to outline some of the key challenges of this process. Before then going into highlighting opportunities for model-informed drug development, obviously that's not the only means that can be used to optimize the drug development process, but it's a very prominent one. And then I believe we'll have time for about 2 examples. All right, so let's jump right in. So first question is, of course, how does a typical drug development process look like, and how are drugs being developed? And so obviously all of you know that we are not giving new drugs or drug candidates right away to patients, but we typically start with a preclinical evaluation followed by an evaluation in animals that is aimed to look at the efficacy and toxicity of the new compound, as well as get a first idea of the exposure response, or so-called PK/PD relationship. So once you basically have narrowed down the list of compounds that you have, and have established lead compounds that have shown have proven to be reasonably efficacious and safe in at least 2 preclinical animal species, then you can move on into so-called phase 1 clinical trials, which are typically primarily geared to look at the safety as well as the PK of these new drug candidates. And these clinical trials are typically done as dose-ranging trials, and they are typically done in healthy subjects. So once you have passed that hurdle, then you move on to phase 2 clinical trials. And phase 2 clinical trials are typically trials that are conducted in patients, where you then look at the efficacy and safety profile at an already narrow dose range. These are typically small to mid-sized clinical trials, and once you pass this phase of drug development, then you go into larger, also so-called pivotal phase 3 clinical trials, who are then also used for regulatory approval and submission, where you test like 1 or 2 dose levels and then evaluate the efficacy and safety in patients in these trials. So once you have completed all of that, you submit the package to the regulatory agency for review, and then you end up either with an approval or a rejection of your compound. But you're not quite done yet, because what then typically happens is that phase 4 kicks in. So these are so-called post-approval trials, and that can span a wide variety of applications, including meeting the need for post-marketing commitments. And this is typically done for pediatric applications, or also if you wanted to establish new indications for a drug that has already been approved. So if you want to put this differently, you go through a stepwise process which is geared to reduce the uncertainty that you have about the dynamic interplay between the drug and the patient. Or in other words, you want to make sure that you stepwise increase your confidence that you pick the right drug for the right patient at the right dose. Please note that there are also deviations from this process. So for example, if you have or if you are developing an oncology compound, then it is obviously unethical to test these compounds in healthy subjects in phase 1 clinical trials. And so typically what you do is that you move ahead into patients in phase 1 clinical trials already. You can also, and this is particularly true if you talk about rare diseases or drugs that have a breakthrough designation, you can also combine different phases of drug development. And so this can be the case for phase 2 or phase 3 studies. And you can also have a little bit of flexibility in terms of when you look at the exposure or the exposure response profile in the studies, depending on the indication that you have. So you can have this in phase 1, 2, 3, or even in phase 4 clinical trials. So as we all know, not all drugs that enter the development process are also being successful. In fact, most of the drugs fail, and I felt that this graph shown here on the left illustrates this quite nicely. And what you see here is the success rate on the y-axis over the development stage on the x-axis. So you have phase 1, 2, 3, regulatory evaluation, and then ultimately approval. But what the bottom line of the graph is that in fact most of the drugs entering clinical development fail. And so if you look at this in terms of the attrition rate, then you see that about 90% of the drugs that enter clinical drug development at the end of the day will fail. So that of course begs the next question, and that is why do these drugs fail? And so if you look at the two pie charts shown here on the right, which give you the reasons for why drugs fail either in phase 2 or in phase 3, that you will see that the number one reason of why drugs fail in clinical development is a lack of sufficient efficacy, followed by safety as the number one reason. All right, so you say, okay, I mean, that's not good. So what does this mean as a financial bottom line? And so I'm showing you some data here that came out of Tufts University in 2014, out of the Center for the Study of Drug Development, which shows that in 2014, it was estimated that the cost for bringing a single new drug to the market was about $2.6 billion. And that, of course, includes the cost that were generated through failed compounds. And so I think it's fair to say that for each drug that you get approved, you have dozens of failures. As a consequence, I think it's also fair to say that attrition in drug development is a major factor in the overall cost of developing new drugs. And so in anticipation that drugs will inadvertently fail during the development process, I think it's also fair to say that if drugs fail, it's better to fail them early instead of late, because that will ultimately result in a reduced waste of time, resources, including, of course, dollars, but also a reduced burden on patients that are serving subjects during this clinical trials. I also think it's fair to say that these data are now already a few years old. So if I had to guess, I would say that today, taking continued attrition, but also inflation into consideration, the cost for bringing a new drug to the market is probably north of $3 billion. That is something that has, of course, also been recognized by regulatory agencies and has also been of concern to regulatory agencies, obviously already prior to 2014. And one of the key documents that came out of, for example, the US FDA was the so-called critical path document. And that is shown here, that appeared in 2004, that highlighted the challenges and opportunities associated with developing new drugs, but also outlined opportunities such as the use of model-based drug development as an important approach of improving drug development knowledge management, but also development decision-making. So what is this model drug or model-based drug development, as it was called at the time, and what is it all about? Well, I felt that this graph here on the right provides a fairly nice overview of what model-based drug development is about. So I think it's fair to say that this is not a single approach, but it consists of a number of approaches that are geared towards evaluating the exposure response, but also the impact of disease on both pharmacokinetics as well as pharmacodynamics. Obviously, it's also important, given that you're not living in a silo once you are in either drug development or in the market space, but you are basically in a dynamic landscape where you have either competitors during drug development or in the market space. So I think it's a good idea to get as early as you can an idea of what the competitive landscape looks like, and meta-analysis has proven to be a very powerful tool in that regard. Model-based analysis approaches can obviously be used to optimize the design and protocols of clinical trials. Obviously, they are also being used to analyze the data that is coming out of these clinical trials. But they can also be used to establish quantitative decision criteria, and we will come back to this in a second, that will help facilitate rational decision-making during drug development, but then ultimately also perform the trial performance metrics in terms of probabilities of failure or probabilities of success. So what that has ultimately led to is a fundamental shift in the way that these trials are being evaluated in a strictly quantitative way, basically going away from a simple focus on beating placebo, because essentially what you're answering is the question, is the drug active or not, towards an ability to differentiate your drug from other drugs. In other words, would it be a viable product in the market space? And so obviously, that is a composite then out of clinical success, regulatory success, but also the ability to get reimbursed for this new drug from third-party parties. And so this probability of success is based on the probability distribution of response that arises from different clinical trials. So how does this ultimately look like? Well, I give you an example here that came out of a publication from Rick Lalonde, former global lead of the ClinPharm group at Pfizer, and now also a member of our team here in Florida. And so basically what this shows here is a variety of things. So let me walk you through this graph in a step-by-step fashion. So what we have is basically two values that are of importance. Number one, and that's abbreviated here with LRV. So that's the lower reference value. So that could either be the placebo treatment or the standard of care. And then you have a target value, and this is for your new compound. And so this target value should be set ideally for both efficacy and safety. So obviously, if evidence arising from the combined evidence arising from a number of clinical trials integrated in a modeling approach suggests that you are exceeding expectations in terms of exceeding your target value here to the right, both in terms on average as well as the when considering the associated variability that would be an indication that you should be going forward with this compound into the next study in the study plan. On the other hand, if the combined evidence from your clinical trials suggests that you are either not beating placebo or the current standard of care, then this would be a clear indication that it's probably a good idea to stop the development of this drug. And that's never an easy decision because people are also emotionally invested in their baby, so to speak, in the compound. But it's probably not a good idea to further invest in the development of these compounds. Obviously, these are two extreme case scenarios, and oftentimes you find yourself somewhere in between. And so, for example, what can happen is that you have a compound where the data that you have generated so far suggests is, yes, that there is an indication that, or a clear hint in the data that this compound is efficacious, but you are battling with a wide range of variability. So you're not quite sure where it will swing. So either, it could swing in your favor, where you then basically meet your expectations and can move on to the next clinical study, or it could swing in a not so favorable direction in a case that you're not beating placebo or the standard of care. So what this probably would tell you here is that at this juncture in the development program, you don't have enough information to make an educated judgment call. It's probably a good idea to pause the development of this compound, generate additional data, and then integrate this into the analysis, and then move forward with making a decision. So obviously, this landscape has evolved since 2007, and I felt that this evolving landscape was very nicely summarized in a post or in a blog from the former FDA Commissioner Scott Gottlieb, where he talks about the innovation initiative at FDA, which is also now part of the sixth iteration of the Prescription Drug Users Fee Act. So it's basically mandated by law. And what you see here, and that is summarized on the bottom right, is that model-informed drug development, as it's called now, no longer model-based, but model-informed drug development, is only part of a bigger machinery where also other factors play a role and are equally important, such as considerations around biomarkers, complex clinical trial designs, real-world evidence, benefit risk assessments, and patient voice. But it is clearly highlighted as a tool that can help to predict clinical outcomes, inform clinical trial design, support the evidence of efficacy and safety, and then optimize the way that we are conducting these and evaluating these clinical trials. So somewhat in parallel, but the concept is the same, we have implemented this, and I would call this innovation initiatives at the College of Pharmacy at the University of Florida. And so this brings me to the examples that I would like to provide today. So obviously, as Dr. Zhao mentioned, I'm in the pharmacometric space. And as I hope I was able to convey. So basically what we do is we integrate data that can either come out of clinical trials, preclinical, or in vitro experiments, and then use mathematical and statistical models to basically predict what would happen in larger patient cohorts, and then predict the response of these larger patient cohorts to a given drug or treatment regimen. Also support the design of respective clinical trials, and at the end of the day, also use covariate relationships to identify subgroups of the population that respond either particularly well to a given treatment regimen or not at all, to make then labeling recommendations. We work very closely with our precision medicine colleagues, so Doctors Julie Johnson and Larissa Cavallari and their teams, where we basically look at the incorporation of genetic or non-genetic sources of variability and their impact on the pharmacokinetics and drug response, as well as then what this means in terms of clinical implications. And we have also started to work very closely with our pharmacoepidemiology colleagues. So this is Almut Winterstein and her team. And conceptually, they basically start the other way around. So they start with the evaluation of very large databases, so what they call the real world data, and then use pharmacostatistical models to basically stepwise reduce the bias that is contained in real world populations to come up with subpopulations that are either responding really well to a given treatment or also can show side effects. And with that, I would like to go into a first example. And the first example that I'd like to provide is an interaction between the pharmacometrics pillar and the precision medicine pillar, as well as the US Food and Drug Administration. And so the question, and that was really a policy example that we had, was how informative are drug-drug interactions of gene-drug interactions. And so you may say, okay, Stefan, that's great, but why do you care? And so the reason why we cared at this point is that regulatory agencies, so FDA, EMA, PMDA, as well as others, expect that clinical pharmacokinetic interactions between an investigational new drug and other drugs should be conducted as part of an adequate assessment of the drug's efficacy and safety. So in other words, you are mandated by law to look at drug-drug interactions if you want to get a new drug approved. And so you say, okay, that's good. But obviously, these studies come at a cost. And so if you look at the cost, then one of those studies costs about $1.5 million. And so the example, or one example that I've seen is the largest numbers of studies being conducted. We were somewhere in the range of about 45 to 50 studies, and you do the math, that obviously adds up fairly quickly. And it's not only the financial investment that you spend on these studies, but it's also the time lost, if you will, during the drug development process that adds up fairly quickly. And so the research question that we had here is, can we use drug-drug interaction data in a reliable fashion to predict G-drug interactions for cytochrome 2D6, 2C9, and 2C19 using prototypical victim structures? And so the research approach that I will walk you through in the following slides was twofold. One, we used a conservative analysis or descriptive analysis that was basically AUC and statistics driven. And the second was an analysis where we used a physiologically based modeling approach. So how did we select the respective enzymes, substrates, and inhibitors? Well, we looked at these three enzymes, so 2D6, 2C9, and 2C19, because they are polymorphic in nature and they have clinically different phenotypes. We selected then prototypical substrate drugs, and given that we were one of the first groups to look at this in this way, we picked, to be honest, a somewhat idealistic scenario. So we picked prototypical substrate drugs that had a fraction metabolized by a single cytochrome pathway. And so for 2D6, this was metoprolol, dextromethorphan, atomoxetine, vortioxetine, warfarin, flubiprofen, and celecoxib for 2C9, and omeprazole and clopidogrel for 2C19. And we also picked somewhat idealistic strong inhibitors, basically in our proof of concept study, that are preferably selective for a single cytochrome pathway. So we selected paroxetine, fluoxetine, quinidine, bupropion for 2D6, fluconazole for 2C9, and then also fluconazole, fluoxetine, and omeprazole for 2C19s. And so we collected then data from the literature for the FDA data bank for poor metabolizers, for gene-drug interaction studies, or for strong inhibitors for drug-drug interaction studies. And then we calculated the exposure ratios in the presence of either drug-drug interactions or gene-drug interactions using the extensive metabolizer data as a reference point. And so we declared convergence between declared conversions between drug-drug interaction studies if the computed AUC ratios were within the 90% prediction interval, so 80 to 125 in most cases. So how does this look like? For PBPK? Well, for PBPK, it was somewhat more complex. So we basically developed separately models for victim drugs as well as preparatory drugs via the integration of their physical chemical properties, knowledge on their metabolic pathways, and then also inclusion of clinical data following IV and oral dosing regimens. And so obviously this took a number of iterations until we were satisfied with these models and called them sufficiently verified. And once we were at this point, we used them then for simulation purposes. And so we conducted two types of simulation purposes. Number one was that we simulated gene-drug interaction scenarios and then compared the results with drug-drug interaction scenarios, or we simulated drug-drug interaction scenarios and overlaid this then with observations from gene-drug interaction studies. And again, convergence was declared if the observations were contained within the 90% prediction. So how do the results look like? Well, I would say they were fairly straightforward for 2D6. So for all tested scenarios, we had conversions for drug-drug interactions and gene-drug interactions for 2D6. The situation was somewhat more complex for 2C9 and 2C19. And so we in part saw a dose dependency. So for example, for fluconazole, so if we looked at the low dose of 200 milligrams, and we did not have a convergence, but if we looked at the high dose, 400 milligrams, then convergence was being declared. The situation was even more complex for omeprazole because we had also a selectivity issue to consider. So convergence was obtained for omeprazole, but not for other proton pump inhibitors. But also convergence was only obtained at a supratherapeutic dose of 80 milligrams, whereas clinical doses are somewhere in the range of 20 to 5 to 20 milligrams. So and these results were really confirmed via the PBPK analysis. So what you see here on the y-axis are the concentrations over time on the x-axis. So the solid green lines are the mean model predicted lines. The dashed lines are the 5th and 95th percentile. And then the observations are presenting then the observation from clinical trials, either from extensive metabolizers or poor metabolizers in drug-drug interaction studies or gene-drug interaction studies. And so as we see here for 2C9, this was somewhat more complex. So for 200 milligrams, the observations were not contained within the prediction intervals, so no conversions could be declared here. But if we increase the dose, then we basically achieve conversions. And so what do we know? Well, we know that gene-drug interaction studies and drug-drug interaction studies are related because they change the intrinsic clearance of both of the enzyme substrate. And so how can these results be used? Well, we can be used, so the results of these studies suggest that we can use drug-drug interaction studies to inform gene-drug interaction labels for 2D6 substrates. So the scenario is somewhat more complex for 2C9 and 2C19 due to a difference in potency and dose of the inhibitor, as well as remaining enzyme activity for lack of loss of function allele carriers. But at the end of the day, these type of analysis may provide an alternative for studying both drug-drug interaction studies and gene-drug interaction studies clinically. In other words, they can be used to save time and development. Dr. Zhao, how much time do we have left?
I'll give you 1 more minute.
Okay, so let me jump ahead to the pharmacometrics and real-world evidence analysis. So we looked at drugs. I provide you here an example for both their application for generic evaluation as well as, so where we basically looked at the impact of biopharmaceutical properties, including particle size as well as formulation properties on the efficacy and safety, and we can use an integrated PK approach here. But what I would like to take your attention to is that we were also able to retrospectively, and maybe that's why I would like to have this here, and to look at prescribability questions. And so we selected DOACs in that regard, and specifically the Vigatron. And again, this is a drug-drug interaction and gene-drug interaction scenario where we have cytochrome-mediated interactions as well as PGP-mediated interactions. And we know also from the drug label that renal function plays a role. So if you look at the Vigatron labeling, then you'll see that if you give it alone, that it's typically given at a dose of 150 milligrams twice daily. If you have patients with a renal function of greater than 30 milliliters per minute, and then the dose is being reduced to 75 milligrams twice daily if you have patients with severely reduced renal function. If you give PGP inhibitors on top, then you will see that dose reduction is also recommended in subjects with a creatinine clearance of 30 to 50 milliliters. So we looked then here into a real-world evidence analysis approach, and this is just 2 slides. But what we found here in this analysis, when we looked at the impact of PGP inhibitors, either verapamil or diltiazem, compared to an active comparator, so in that case with either amlodipine or metoprolol, that the hazard ratios for experiencing bleeding events were significantly increased for the Vigatron as compared to rivaroxaban or apixaban, and that was published in JAMA, suggesting that a dose should be reduced in subjects also with normal renal functions receiving PGP inhibitors. And so that, of course, begs the question, what do you do in subjects that have renal impairment on top? And so we worked together with Jomi George from NIH, looking at data from people with living with HIV receiving booster drugs, including ritonavir and cobicistat. And so basically what we used in the modeling and simulation approach for is to simulate out our expectation for a variety of dosing regimens, either the standard dosing regimen or then stepwise reduced dosing regimen, either given concurrently or dose separated. And what we see is that the impact is different for different PGP inhibitors. So it's more pronounced for cobicistat. And the bottom line here is that the Vigatron should be avoided in subjects with severe renal impairment, either receiving ritonavir or cobicistat. Subjects with moderate renal impairment receiving ritonavir, so the dose should be reduced, should be either separated if possible clinically or reduced to 110 milligrams twice daily. If you have moderate renal impairment for cobicistat, the dose should be reduced to 75 milligrams twice daily. No dose adjustment is necessarily in subjects with normal renal function with ritonavir, but it should be reduced for cobicistat. So bottom line, drug development is a complex process. No single experimental or clinical trial or analysis approach can answer questions. However, the complexity of the question should drive the complexity of the analysis or the study, not the other way around. And the integration of knowledge is key. Obviously, we needed to have financial support, and I'd like to thank all the sponsors as well as the research team. And with that, I'm open for questions.
Yeah, thank you very much, Dr. Schmidt. Probably because of time, we probably can only answer one question. I got the first one from Dr. Liang. Asked your comment, to what extent FDA accept DDI data from in vitro and animal study in replace of actual human clinical trial phase 1 PK-DDI study?
This is a great question. There is a clear trend in FDA that the integrated data, so the integrated data from in vitro and animal studies as part of a model-based package, for example, using physiology-based modeling approaches, is routinely accepted as a surrogate for clinical trial. And so that has basically become standard right now. In fact, if you take it one step further, the EMA has basically now also proclaimed that it wants to do away with animal studies by the year of 2030. And so the integration of in vitro and animal data, or also human on the chip or organ on the chip data, together with some modeling approaches, the clear avenue.
Okay, we have a few more minutes. Maybe we can entertain the second question from Dr. Xie. She would like to know your comments on FDA's fast track for drug approval in the current pandemic situation.
Yeah, so that's a great question. I mean, obviously, there is a need to expedite the drug development and regulatory approval process while making sure that efficacy and safety are not being jeopardized. And I think both FDA as well as pharmaceutical industry is trying to employ the utmost rigor in the process, as evidenced by the fact that if you look what's happening, for example, from AstraZeneca, that they're holding clinical trials if signals pop up. I think that speaks to the rigor that's being applied. At the same time, you want that rigor to be applied in order to have also at the end of the day confidence by the end user, so the patient, that the medication is efficacious but also safe. And so I think there is it's a tricky situation to work in this field both at FDA as well as pharmaceutical industry, but I have the utmost respect of the work that's being done there.
Thank you very much. For whatever the question, if you still want to ask, you can enter into the chat, and Dr. Schmidt will try to answer through there. Okay, thank you very much.
Our next speaker is Dr. Song Gao from Texas Southern University. Dr. Gao.
All right. So good afternoon, everyone. So today I'm going to use a project to demonstrate our capability of doing in vitro absorption, drug absorption, and metabolism in the GCC separate center. So irinotecan is a chemotherapeutic drug used for the treatment of different types of cancers, but the challenge is late onset diarrhea. So it is reported that more than 80% of the patients experienced diarrhea when treated with irinotecan, including 40% up to 40% of severe diarrhea, which is grade 3 and grade 4 diarrhea. So the mechanism of diarrhea is not entirely understood, but it is well known that this is a dose-limiting side effect. So different approaches has been recommended, but unfortunately, a lot of patients do not respond well for this non-pharmacological and pharmacological treatment. Then we have to reduce the dose to manage diarrhea. But as you know, that when the dose is reduced, the efficacy is going to be affected. So since this is a dose-limiting side effect, so the drug is in vivo exposure is critical. Irinotecan actually is a prodrug of SN38, so it can be converted into SN38 through carboxylerysteresis. Unfortunately, SN38 is also the toxic compound. So SN38 can be converted into SN38 glucuronide mediated by UGT1A1, which is a FIS2 metabolism enzyme. So this three compound also can be secreted, can be effluxed through different efflux transporters like BCRP, MRP2, and PDP. So this is a major metabolic pathway. So the CYP reaction is a minor pathway. So if you will look at the disposition, these three compounds participate in intrahaptic recycling. And then locally in the intestine, a lot of factors are involved in the to control the intracellular level. So it is believed that SN38 can damage the gut cell to cause diarrhea. So the question is whether the SN38 exposure level in the intestine is correlated with diarrhea severity. So to answer this question, we developed an animal model by injecting irinotecan to mouse for 6 days, and then we collect the feces and evaluate the fecal condition. So the model is successfully established using different strains of mouse. For example, in FEB mouse, on day 6, there are 20% of grade 1 and 10% of grade 2. And on day 9, there are 40% of grade 4, 30% of grade 3, and then grade 2 and grade 1. So in this model, irinotecan is given by IP, which is different with that in human. In human, irinotecan is given by IV infusion. Then we establish a IV infusion model using rat because mouse is difficult to do IV infusion. IV bolus is fine, but IV infusion is difficult because the duration should be 30 minutes or even 1 hour. So we inject irinotecan in BCRP, MRP2, and the BCRP knockout mouse, and we found that the diarrhea severity is different because these three transporters are involved in the drug disposition. And then we quantify the drug concentration in the intestine and also in the plasma. And we found that intestinal SN38 exposure is correlated with diarrhea severity. If you look at this figure, I mean, when the intestinal concentration of SN38 increased, the severity of diarrhea is increased, which is proposed in the literature. Then our goal is to selectively reduce the intestinal SN38 exposure for diarrhea attenuation, because we don't want to affect the systemic SN38 exposure, which is associated with therapeutic efficacy. Now, if you look at the local disposition, a lot of factors are involved as we showed before. So a lot of previously effort has been attempted to reduce the intestinal lumen concentration, which is a donor to the intracellular SN38 exposure. Then we measure the permeability of SN38 using a CACO2 cell culture model. In this model, the CACO2 cells, which is a colon cancer cells, are seated on an insert to form a monolayer. Then we upload the drug on top of the monolayer, which is called an apical site. And then periodically we take sample from the basolateral site, and then we quantify the drug concentration to determine the uptake permeability, or the other way around to determine the efflux permeability. So this is a FDA recognized model to determine the drug permeability. Then we found that for both irinotecan and SN38, the permeability from B2A, which is the efflux permeability, is significantly higher than uptake permeability. In other words, while the donor concentration, if you decrease or increase the donor concentration, the impact on the intracellular level is low. Since the CE, expression of CE in the intestine is very low, then we hypothesize that intestinal UGT is the factor that dominated the intracellular level of SN38. And also, we hypothesize that if we can selectively induce the local UGT, we can decrease SN38 level in the intestine for diarrhea attenuation without affecting its efficacy. And now to study the UGT, or UGT1A1 is the major isoform to metabolize SN38. So we develop a LC-mass method to absolutely quantify UGT1A1 at protein level. Then we sacrifice the animal on different days, and then we collect the tissues and measure the UGT1A1 expression. Interestingly, in the intestine, the UGT1A1 expression level gradually decrease as we inject irinotecan. Surprisingly, in the liver, the UGT1A1 expression didn't decrease. So we don't know the molecular mechanism so far, but this UGT regulation is organ dependent. So the next question is, which compound regulates UGT1A1? So we incubate SN38 and irinotecan with CAC2 cells, and we find that SN38 downregulates UGT1A1 at a nanomole range. Irinotecan didn't, even at a micromole range. So we can conclude that intestinal UGT1A1 expression was downregulated by SN38. This is the expression level. So what is the functional level? And then we sacrifice the animal on different day, and we collect the tissues and homogenize and centrifuge to make a S9 fraction and then microsomes. So this is a standard procedure to prepare enzymes. We use this procedure, we can concentrate the enzyme. And then we incubated the enzyme with SN38 and quantify the SN38 glucuronide using UPLC, and then calculate the metabolism rate. So in the intestine, we can find that SN38 glucuronidation rate at a different day, gradually decrease on different day. So which is not surprised because the UGT1A1 expression level decreased. And then in the liver, there's no decrease. Then we look at the drug exposure. We sacrifice the animal at a different time on day 1 and day 6, and we collect the tissues to measure the SN38 exposure. And we can see here the AUC on day 6, SN38 in the intestine increased dramatically compared with day 1. In the liver, SN38 exposure didn't change. So remember, this concentration is correlated with diarrhea severity, and also the liver concentration is correlated with plasma concentration, which is associated with efficacy. Now we are confident that we should identify an agent that can induce or protect intestinal UGT so that we can decrease the intestinal SN38 exposure for diarrhea attenuation. And then an herbal formula, which is called XCXT, comes up to our mind. This herbal formula has been used in China and Japan for hundreds of years without a severe toxicity reported. It had 3 clinical trials in US, but it's used for different disease. And so this formula contains 7 herbs. Like ginger, like a jujube. So I eat it like or in my cooking, or eat it as a as a food. So the safety is not a problem. And the next question is, what is the function, the impact of HCX on the UGT expression and activity? And then we give HCXT together with irinotecan and sacrifice the animal on different day. And we found that a UGT1A1 expression in the intestine didn't decrease when the animal was treated with HCXT. On the functional level, with HCXT treatment, while the liver UGT activity increased 2 to 3 folds, but if you look at the intestine, the UGT activity increased dramatically. On day 4, it's like 120 folds. And then we look at the drug accumulation. We sacrifice the animal on day 6 again, and with HCXT treatment, so you look at this red line, so the SN38 exposure in the intestine decreased. This is the AUC. So remember, this is correlated with diarrhea severity. And in the plasma, SN38 concentration didn't change, which is associated with efficacy. And now we can conclude that HCXT impact intestinal UGT selectively, which is our expect. Then what is the impact of HCXT on the biliary secretion? So we determine the biliary secretion using a in situ intestinal perfusion model. So this model is also a standard model to study drug absorption. Usually in this model, the drug is perfused through, so we cannulated the segment of intestine and also the bile duct. And then the drug was perfused through the intestine. We collect the perfusate and also the bile, so we can quantify the drug concentration in the perfusate and also the bile and determine the drug absorption and also the biliary secretion. So in this experiments, we modify this model by infused irinotecan through the jugular vein and then collect the bile to determine the biliary secretion. HCXT was perfused through the intestine. And the result showed that in the presence of HCXT and the biliary secretion of SN38 decreased. Well, the next question is whether HCXT can against diarrhea. Then we give the animal HCXT at different dose together with irinotecan. We find that HCXT against attenuated irinotecan-induced diarrhea following a dose-dependent manner. This is in a mouse model. Then in rat, we also find that HCXT can attenuate irinotecan-induced diarrhea. While this model is, while the drug administration route is similar as that in human. So the next question is the efficacy, the impact on potential impact on efficacy. So we give HCXT at a different dose together with irinotecan in a xenograft model, and we found that HCXT did not affect irinotecan's therapeutic efficacy, so which is very good. Then comes to our conclusion that intestinal SN38 exposure is correlated with diarrhea severity. The SN38 downregulates intestinal UGT expression to cause late onset diarrhea. And HCXT can selectively induce intestinal UGT reduction caused by SN38 to attenuate diarrhea without affecting its therapeutic efficacy. So I appreciate the hard work from my staff and students, and also I appreciate my collaborators, Dr. Ming Hu and Dr. Ghosh at UVH. Actually, a lot of work were conducted in their lab. I have been cited the publications in my slide, and also I appreciate the grant support. All right, that's what I have today. I'm very happy to take any questions.
Thank you. Thank you, Dr. Gao. I guess we are running a couple of minutes over. Probably, Dr. Gao, you may have to stay on to the ballot if there is more questions on that.
Sure.
That way, we're probably going to go ahead and close the session.
Sure.
Suzanne, do you want to make any comments?
Sure. We certainly want to thank everyone for being here today. Definitely thank our speakers, the conference organizers, core directors, everybody who's contributed to today. I definitely want to invite the speakers to continue to respond to questions in the chat, and also our audience to continue submitting them if you have additional questions. We've kind of tagged this last bit of the day for networking, which certainly looks a lot different since we are in a virtual reality right now. But, you know, it's really important that we try to be creative and continue to find ways to collaborate, meet new folks. And I really want to encourage you to look through the list of conference attendees on your Umbrella interface. You can click on them and request a meeting. You can actually jump into a video chat with them right in that window that it pulls up. We will leave this up through the day and even tomorrow, but we hope that for the next hour or so, you stay on and reach out to some folks, make some new connections, and hopefully they'll lead to some important collaborations to advance your research. Again, thank you to all, and we look forward to seeing you next year.